All right, so, oh, this is not what I want. That's also not what I want. That's what I want. Okay, 2025, Semester 1, SBI3U 3C, Unit 1, Lessons. There we are. Sure. All right. So again, sorry I'm desk bound here today. I feel very weird not being up and about, but I didn't charge my computer, so if I unplug it, it'll die. Okay, eukaryotic development. Remind me, what are eukaryotes? What are eukaryotes or eukaryota? Yeah. Okay, complex cells with nuclei and with organelles. That rhymes, which is enjoyable. Got sort of an... Yeah, it's got kind of a cadence to it. Anyway, complex cells with nuclei and organelles. We said life on Earth is about 3.5 billion years old. The Earth itself about 4.5 billion years old. Our solar system about 5.5 billion years old. Nice easy numbers there. We said for the first few billions of life on Earth, there were only prokaryotes. Scientists are not entirely sure how eukaryotes even developed, but there's one major theory that's often cited. All right, one major theory that we most often tend to think explains the rise of eukaryotic life, which remember means life that has organelles inside the cells, right? So how's that work? What's that look like? Well, let's imagine before we get to it that we could break this word down a bit. Endo and symbiosis. What's endo mean? Endoskeleton, endo, inside. Very good. Endo means inside. And what's symbiosis? What's symbiosis? This is just an English word. Though it actually comes from two roots. In fact, ready? Let's do this. Sim as a root. Symbiosis, sympathy, sympatric, simpatico. Together. Sympathy comes from the word sim, meaning together, and pathos, meaning suffering. If you have sympathy for someone, you're suffering together, or you're suffering with them. Symbiosis. Bio, of course, is, which is life. Inside together life. Inside together life. What's that mean? But a symbiote is what? It's something that lives inside alongside with you. Does that make sense? Not necessarily inside. The inside is here, but you live symbiotically if you live alongside each other together. You have a symbiotic relationship with something if your life depends on them and their life depends on you, right? Sim together life. Life depends on each other. Animals and plants have a symbiotic relationship. What do plants produce lots of? What do animals require lots of? Oxygen. See, that's symbiotic. They, on the other hand, require lots of carbon dioxide. What do we produce? Carbon dioxide. Does that make sense? So we live in a symbiotic relationship where we replenish one another's. Oh, just let me know when it's good. It's now good?
4 minutes maybe?
How about now? Does that work? That works for me. Settle down. All right, so here's the basic idea then. The basic idea is that you can imagine a simple eukaryotic cell All right? And it's got its little loop of DNA. What do we call that little loop of DNA? Oh, not eukaryotic, sorry, prokaryotic. What do we call the loop of DNA inside a prokaryotic cell? Anyone? Plasmid, right? Remember we talked about that briefly when we talked about the lytic cycle. And as it happens, this is a hungry little cell. And we've got this little cell here, and he's living there, and he's got his little plasmid of DNA. And this is another prokaryotic cell. And sometimes, when a big prokaryotic cell is hungry and a little prokaryotic cell is around, what happens? Yeah, it's going to eat it. It's going to engage in a process called pinocytosis or phagocytosis, whereby we are going to engulf the little cell. This is the thing that can happen. And this is often how a big cell will feed on a smaller cell. It will engulf it like this, where the membrane will surround it, and then normally it would bring it inside, and then what would it do with it? Well, but what would normally happen if we're feeding on something? We'd take it inside and we would... Yeah, we would just take it right apart and we'd steal all its nutrients, right? Does that make sense? That's what you do. You put other living things inside your face and you chomp on them with the big, sharp, grindy teeth ears, and then you swallow them and then you steal all the nutrients. So, what can happen is big cell engulfs the small cell, to digest its nutrients, right? But then something happens. And the thing that happens, we think, in the past is kind of interesting. And the thing we think happened in the past that is kind of interesting is that instead of dying out, we think that that inner cell with the nucleus, and I should have made the DNA in this DNA colored, which in this case I guess means red the whole time. We think that we got kind of this situation where the smaller cells survived inside the bigger one. And then over generations, the outer DNA was able to degrade or was able to make its way into the inner cell. But in one way or another, eventually all of the DNA, the only DNA, was found inside the inner cell. And what do we end up having there? And what we have there then is the development of the nucleus. I wrote that a little big. If you also wrote it a little big, what you could do is just kind of highlight that like this with your pen, and then you could shrink it a little bit and kind of slide it over a little bit.
I would love to. Also, they look like olives.
It does look a little bit like an olive with a pimento, doesn't it?
Yeah.
So this process could explain how the nucleus developed. And again, you have to think, why would the DNA inside the nucleus survive while the DNA in the cytoplasm didn't? Well, we've always known that the cytoplasm is sort of a dangerous place for DNA. Bacteria undergo all sorts of mutations and change sort of the genetic material quite frequently because their DNA is exposed relative to ours. So this theory states that eventually this would serve as a way for the DNA to be more protected than in either of the original prokaryotic cells. So you end up with this symbiotic situation where you have essentially a cell living inside of another cell. But that inner cell we call an organelle. And the nucleus basically is that. It is a cell inside of a cell. It's surrounded by a nucleus, and it has the DNA inside of it. And in addition to the DNA, it also has all kinds of proteins that do a bunch of work in there, that wind the DNA up, unwind it, help it reproduce, help read it, and get certain areas. Yeah, what's up?
Wow, how interesting. Okay, I just sent you, did you order me those calculators last year?
Yes.
You did?
Yeah.
Oh, you did?
I believe so.
Okay.
I might be wrong.
Okay, if you
could
didn't-
be out of budget. How many do you want?
Well, just a sec.
And these are the ones?
Tim ordered these for Courtney.
Okay.
This is all I need for my little guys.
Perfect.
Amazon, she's gonna send me the link. If you didn't order me some, just maybe like
Yeah, I can't
around
remember if we were out of
20.
budget or not. I'll check 20's good.
Is that okay? And then just these cheapos.
Yep, beautiful.
Okay, sorry to interrupt you.
No, no
I'll,
problem. You'll send me the link?
yeah, thank you.
Great. Yeah. What does what say? This
This process.
could explain the development of the nucleus. Sorry, it's like half cursive. I do that sometimes. I get a little half cursive. Sorry? You don't need to ask me that. You good to go? In other words, a way to think of the nucleus is as just a cell inside of a cell. That is where our DNA lives. Now, what other cells live inside of our cell, so to speak? What are the other organelles? Mitochondria. So the mitochondria would be a good example where if an early cell absorbed another cell that used sugars and produced energy at a very high rate, that cell may have survived and become the mitochondrion. And it's the mitochondrion that actually gives us the best evidence for this having happened. So you can imagine, by the way, we don't claim to know what order these happened in. So it's possible the mitochondrion develops before the nucleus. But I'm going to try and kind of draw it here like it did, like it developed second. So I'm going to say, okay, we've got our cell here, and it's already got a nucleus, and that nucleus has some DNA inside of it. So there we are. And it stumbles across this other cell, which we always seem to draw in red, and we always seem to draw with its DNA like that, because that's what it looks like under a microscope. It's got this, or not DNA, it's inner membrane. It has this highly folded inner membrane that's used to burn sugars. You kind of draw it like Pac-Man. It's kind of like a really like low-rent, crazy-eyed Pac-Man, but there we go. It still makes me happy. And what we end up with is a cell that has... I'm going to cheat and make the DNA blue so it's not the same as the mitochondrion. Try and stop me.
Is it used? Used or used?
Where?
Up top. Oh, used, okay.
Yeah. But this actually gives us a little evidence for this. The evidence that this may have happened is that, and you might know this, you your mitochondria have their own DNA called the mitochondrial DNA. And you'll be shocked to hear, what does the DNA inside the mitochondria code for? It codes for all the stuff in the mitochondria, all the stuff that breaks down sugars. Bless you.
Thank you.
Bless you.
Thank you.
And the neat thing is, that's actually entirely independent of the DNA in the rest of your cell. We know this for a few reasons, but one that makes it really interesting is, where does most of your DNA come from? It's in your nucleus, and where do you get the DNA in your nucleus? From your parents. Which parent? That's right. Mother. Does your mother just give you your DNA? What about your dad? And they both give half and half, right? That's how fertilization works, right? We say that humans produce sexually and animals reproduce sexually. And so you have a sperm cell, which is a sex cell from dad. You have an egg cell, which is a sex cell from mom. And when they come together, they both give you half of your DNA, right? But interestingly, your mitochondria, which cell is sort of the proto-cell that becomes the embryo? That has all the stuff it needs in it? The sperm or the egg? Which one's sort of the primary cell that, ends up in the zygote? Do you know? You just look at the size of them. Which one's bigger? Yeah. The egg, right? The sperm basically just provides DNA from dad. But the egg has all the other stuff that's going to grow into your cells, which means that the egg already has mitochondria, endoplasmic reticulum, all these structures that we need. And what's interesting is your main DNA, the DNA in your nucleus, you get half of it from mom and get half of it from dad. Your mitochondrial DNA is matrilineal. What's that mean? Matrilineal. 100% from your mom. Because the mitochondria were already in the egg cell that comes from your mom, and that's what reproduces to form the mitochondria in your new cell. So we don't talk about it as one of our steps of mitosis, but one of the things that happens during interphase is the mitochondria reproduce themselves, and when the two cells split in half of them get the mitochondria, half of it on one side, the other cell gets the other half of the mitochondria, but those have been self-replicating, and that DNA comes from the egg cell that your mom provided. So you can track somebody's DNA up their father's line and their mother's line using the main DNA inside their cells. But if you look at the mitochondrial DNA, it's matrilineal. All you're looking at is your mother, and then your maternal grandmother, in other words, your mom's mom, and then her mom, and her mom, and her mom, and you can actually track the lineage of a person just through the mother's family line by looking at their mitochondrial DNA. We'll use mitochondrial DNA in a lot of analyses of things, because sometimes it's better preserved than nuclear DNA is, and sometimes it's not. Does that make some kind of sense? Cool. But that actually serves as a piece of evidence that this process is likely what happened. Likewise, the chloroplast, in this case, the theory is that a cell that could photosynthesize was absorbed by a cell that couldn't, and now it was a cell that contained an organelle that could do photosynthesis. So a eukaryotic cell likely absorbed a photosynthetic prokaryote. That's an R.
Oh.
Sorry.
It's okay.
So now we have a cell, and it's got mitochondria throughout it. Oh, there's another mitochondrion. There's a mitochondrion. There's a mitochondrion. And it's got the nucleus. And by now we're deep into development, so we no longer have a plasmid. We've got that there. And we come across this cell here that is very good at photosynthesizing. You'll always see it drawn with these little structures called the thylakoids, which don't worry about. You'll learn about in grade 12 if you care. And endosymbiosis happens. And now we have modern photosynthetic cells, where you have that, you have the nucleus with all the DNA in it. We've got the mitochondria, right, there they are. And now we've got this other structure called the chloroplast. Ask a question.
Yeah.
Do you appreciate or resent that instead of just giving you the diagrams, I draw them and make you draw them? I think it's helpful. I think it's helpful, and I tell you this as somebody who you'll be shocked to hear me say I don't think I'm very good at drawing, but I still think that there's a lot of value in it because it helps you sort of, when you have to draw it, you have to make decisions of like, how big is this thing? What color is it? Where does it go? And that makes you sort of really consider the diagram and remember it.
I like the little drawings.
I practice them. When I have a lesson where I know I'm going to have to do lots of drawing, I practice the night before and I get all stressed out. It's a true story. Art causes me stress. It doesn't come naturally to me.
It's like the opposite.
I know.
Yeah.
One of these days I'm going to like get you a second tablet on the same workbook, and every time a drawing comes up I'm going to be like, go! And you can just do it.
We'll just be we'll have to delete some of these. Yeah.
Alright, do you guys need a minute? No,
Can you do attendance?
I suppose.
Just
Yes,
My mother
ma'am.
called me the other day saying that wasn't in your class.
Were you away?
No.
Well, that isn't because I didn't do attendance. Because interestingly, that when you don't do attendance, it assumes everyone is here. That just means I missed you, which I apologize for. Are you sure you weren't here? Or you were here?
I was here. And I got called and my mother asked where I was.
Skipper.
I don't skip.
Britton's here. Becker's not here. Evan's here. Evan? Ross? Oh, right here. Literally in my line of sight, like right there. Deanna's here. Saphira's here. Audrey, Kayla, Aiden, Kira, Kardik, Andrea, Angela, Annie, Maggie, Samuel, yeah. Ellie's not here. Kashi's here. Solange is here. Sunny is not here, right?
Mm-hmm.
Okay, perfect. All right. There you go. Attendance done. Are you happy?
I won't get yelled at.
I mean, I'll still get yelled at, but it'll be something different, so that's good. I like to mix it up. Okay, so we're gonna really quickly look at me, give me 15 minutes, and after that, tell me to be quiet and let you guys get some work done. We're gonna quickly look through just some examples of this. All right, so multicellular and unicellular organisms are both possible when you're talking about eukaryotic cells. This should start on the next page for you, does it?
There we go, yeah.
Yeah, okay. I don't know why mine it didn't. It does that sometimes.
Why is there just a white page?
Sorry? I don't know. What do I look like? All right. What are we looking at here? This is an electron micrograph. This is just a typical a light microscope. But what are we looking at here? Yeah, this is Saccharomyces cerevisiae. Does anyone know what Saccharomyces cerevisiae is? Yeah.
Not exactly, looks like used in bread and wine and stuff.
Yeah, very good. So this is one of the most useful organisms on planet Earth. We use it to make bread. Has anyone ever made bread before? If you add yeast to the dough, what does the yeast do? Yeah, it puffs it up. Very good. Do you know why? Because the yeast is a living organism, and what you're doing is you're putting it in a big pool of like nutrients that it can eat, and it feeds on it. And while it does that, it produces carbon dioxide, which gets released as a gas. But because that gets trapped in the dough, it makes big, like, puffy cabinets. But don't worry, then you put them in the oven and you kill those yeast real good by baking them. After they've helped you, you're like, haha, take this yeast.
They explode too.
No, we trap and
But somebody mentioned wines. Saccharomyces cerevisiae, you think we were mean to them baking, where first we gave them a bunch of delicious food and let them produce a bunch of carbon dioxide, then we baked them. In wine, we give them a bunch of delicious juice, and then we don't give them any oxygen. We trap them in there. We trap them in there real good. And so they panic, and they start doing a process called fermentation, which is like panic survival, where they can burn those sugars at a big energy discount, but it produces a waste product called ethanol, better known by just its pejorative name, alcohol. And we like this because it's a toxin. We know it's a toxin because it kills the yeast. They produce this ethanol, and it kills them because it's poisonous. And what we do is we like to drink that on purpose, and it causes you brain damage. It's called being drunk, and people like this too, because we just drank intentionally a toxin so we can do brain damage. But don't worry, if you're wondering, is it also a class 1 carcinogen that does systemic damage to all of your organs, and is it correlated with extremely high rates of kidney failure, liver cancers, pancreatic cancers, stomach cancers, urogenital cancers? Don't worry, it is. If it were any other product except for something baked into human, if like a new product came on the market that caused as much cancer as alcohol, they would never clear it for market, unless it was called vaping, which is a different discussion. But alcohol, incredibly terrible for you. Terrible, again, poisonous to the yeast too. But we starve those bad boys of oxygen real good so that they produce it for us. You can draw your own conclusions about the morality of that. I used in beer making. Yeah, so Saccharomyces cerevisiae. But Saccharomyces cerevisiae is a single-celled or a unicellular fungi. Most yeasts, like Saccharomyces cerevisiae, reproduce asexually by budding. Budding is a little bit different than binary fission. What's the difference between this and binary fission? When we watch binary fission, what do we expect to see happen? That's how bacteria reproduce. It's also different than mitosis, which is what we expect. Why? How does this look different? Yeah, they bunch together, but they'll eventually break apart. What I note is that, do these two cells look identical right now? Eventually they will, but with binary fission and with mitosis, as soon as it happens, the two daughter cells look identical to one another, right? And there really isn't a parent cell left behind, right? But here, what happens is the parent cell doesn't get cut in half. It starts a little bud here, and it moves half of its DNA into it, like it duplicates the DNA and moves half into it, and then that can grow from there. Does that make sense? So the primary difference between budding and binary fission is that the developing bud starts out small, gets its DNA, and grows in place while separating off. Does that make sense? So it gets DNA, grows, and separates, right? As opposed to the cell grows and then gets cut perfectly in half. I mean, not perfectly, but roughly in half, which is how it happens in binary fission. Does that make sense? I don't want to go into that in too much detail. This isn't a fungus course. I took 2 full years of fungal mycology at Brock University. It was fascinating, great courses, would recommend. We got to go to a mushroom farm.
Ooh,
Do you know at the time mushrooms were the second biggest crop farm in a cash crop in Ontario? I don't know if that's still true. Isn't that wild? Huge cash crop mushrooms. They don't grow them outdoors. They grow them in these big, like, just, yeah. Sorry? Sure? It's like, if I turn off the lights, I think you're just boring enough that I could nap. I've done the math. It's yeah, there you go. Oh, jeez. Spooky. Should I tell like a ghost story?
Sure. Sciencey ghost story.
Yeah. Let's quickly talk about the yeast cell. We don't need to get into it too much, but the main thing to note is the eukaryotic. So they have all of the expected organelles. But the other thing to note is that between the membrane, or outside of the membrane, we have what's known as the periplasm, and we have what's known as the cell wall. So there's a cell wall around the membrane with a layer of fluid between the two called the periplasm. Peri is a prefix that means all around. You might think of, they talk about prenatal care, which is the care a mother gets before the baby's born, and antenatal care, which is the care a mom gets after the baby is born. And they talk about perinatal care, which is just all of that put together. Peri means sort of in total or all around, and plasm refers to fluid. So it's the all-around fluid. It's a good name, right? Again, if you speak Latin, these names aren't fancy, they're just yeah, we won't get into too much detail, but then we'll get into multicellular fungi. And this is what you'll see a lot of. Surely you've seen growths like this on things before?
Yes.
This is like a fungal growth, a mold growth, right? What does it look like when something gets moldy?
Which kind? There's a lot.
Yeah.
It's kind of like furry.
Yeah, it gets that kind of furriness to it, right? Has everyone seen that before? That kind of gross furriness on a piece of bread or a piece of fruit or something like that. You ever eaten a piece of bread and then you look at the bag of bread and one of the pieces in the bag of bread has some mold on it, but you just ate a piece of bread? That ever happened to you?
No.
That happened to me like a week ago and I was so horrified. I was like, did I just eat moldy bread? And then it's like, there's only 2 moldy pieces in the bag. Did this one have mold on it? I don't know if this one had mold on it. Ruined my week.
Is your week
Yeah.
don't all the pieces technically have pieces of mold just a network of fungi?
It's true, but yeah. So, but you know, you don't want it. By the time it's visible, you're not interested, right? This is the thing, right? We can all agree on that. Now, mind you, this is like, you know, how drugs were discovered. This is like how penicillin was discovered. But what is that furriness? If you actually look under a microscope, it's this, which is what a fungus, a multicellular fungus, typically is. Their cells are these long, slender tubes that are interconnected, and that we call those hyphae. So each individual cell is called a hypha, plural hyphae.
Where do you even see?
Sorry?
I don't understand what you're seeing.
Do you want to hold up your page?
Oh, it's on the.
Over here.
There's a blank page for some reason.
Oh,
Like
sorry
on
about the
front.
blank page.
Yeah,
I didn't mean to do that.
So it's on the back of the
So if you
page.
look here, inside these hyphae are the individual cells. So a single filament, an individual filament is made up of multiple cells, as seen below. But these will grow longer and longer and longer as new cells form. And a new cell will form here, and now you have a longer hyphae, and so on and so forth. We call the network of these the fungal mycelium. The network of hyphae is called a fungal mycelium. This is the actual organism. When you think of the most common sort of fungus, what do you think of? People always seem to think of the same thing. What's the fungi everyone kind of knows? Delicious on pizza. Mushrooms, right? What's a mushroom? Sorry? Well, what part of a fungi is it? It's like the naughty bits. It's a fungal sex organ, right? That's what and don't feel bad. That's what flowers are too. That's a plant's naughty bits, right? But when you see mushrooms growing in a field, that's not the whole organism. That is just its fruit, what we call the fruiting body, but that's just the sex organ, right? The actual fungus under the ground, anywhere you see a mushroom, if you dig in the ground, you will find this just furry network of filaments running through the soil. That's the fungus. That's the mycelium. Does that make sense? The mushroom sprouts above ground for reproduction because what does the mushroom produce? Just like the plant grows fruit because the fruit contains the seeds. In a fungus, what does the mushroom contain? Do mushrooms have seeds? Do fungus have seeds? Fungi have seeds? Is that a hand? Oh, $1,000 answer. Fantastic. I don't actually have money, just so you know. Yes, spores, which is the equivalent of seeds for fungi. So, if you look here, we get within the hyphae, all right? So if you look here, we get the hyphae growing. So this is the main body of the fungus. Begins to grow. Now, you don't need to know this in detail, but they begin to produce these things called a sporangium. And that's more than you need to know, but that's okay. The sporangium will eventually grow into this structure that we more commonly call a mushroom. But a mushroom is an example of a sporangium, or a sporangia. And when they burst open, the gills of a mushroom contain fungal spores, which are comparable to seeds. The difference is they're very, very small and very, very light. What's cool about fungal spores is I read once about an experiment they did where in North America, like up in Saskatchewan, they took some mushrooms and they irradiated those spores with a distinctive pattern of radiation, so they'd be able to find, if they found them somewhere, they'd be able to know that it was from this one plant. And they let these spores blow away like they normally would in the wind, and they were finding them as far as like South America and Mexico. They're so small that the wind can just catch them and they can go. Or even better, they're so small that they can end up in a bird's feathers or something like that, and they can just end up halfway across the world. Does that make sense? So fungi are very adept at procreating over long distances. Yeah. Sorry? This way? How's that? Again, we're not going to go into great detail, but when we do our culturing activity where we prepare plates and culture on them, you're going to see that furry, fuzzy network, and then when we look at it under the microscope, you're going to see those individual fungal hyphae. In fact, this photo here was taken with this microscope right here in this classroom from a sample that a student took, which is pretty cool. These ones are not. They were taken from Google Images. All right, and then I've left you with a little homework. Now you may have noticed that this was called Fungi, Animals, and Plants. Are you ready for the animals and plants portion of the lesson? Look at me. Animals and plants also exist, and they're eukaryotic. And and we're going to learn all about them in their own unit. Okay, that's the end of my lesson. Listen, you've got 28 minutes remaining. What can you do with that time productively?
Dichotomous key.
Dichotomous key, what else? When's that due? Tomorrow. What else can you do?
Bacteria, and virus.
Bacteria, virus, and protist disease assignment. When's that due?
Tuesday.
Tuesday next
Not
week.
today.
Cool. What else could you be doing?
Safety test.
Safety tests. When do they need to be done by?
Friday.
No.
Or is it not Friday?
No. When?
Tomorrow?
Tomorrow.
Oh.
Good. The other thing I want by tomorrow is your parents to have signed the safety contract. It's in School Cash Online. If your parents log into School Cash Online, they may have already signed it. They can do so. Not everyone is tech. Does anyone have a parent who maybe aren't the best with tech? Yeah. So if that describes you, you may want to grab a paper copy here. I can take a paper copy as well. When do I want those done by? Tomorrow. Don't mess around with me, team. What's up? What can I do for you?
Am I do my model with 3D printer or like?
Yeah. So what do you? So how are you? Where are you gonna make it?
Oh, what do you mean?
So you got to make it with a 3D printer.
Yeah,
So, do you have your own computer?
Right now, no.
That's okay. You can use mine back there.
Okay.
So we're gonna use a piece of software called Blender. Is probably the easiest thing to make
Yeah.
it with. And in Blender, you can.
I can design it and then see if I can. Then I would print it. But if I cannot, then I can just make it with spaghetti or something.
Yeah, sure. But I think you can do this. You're very smart. Because with this, you can just say add, and we're just gonna add a sphere. There's my sphere. Okay, so there's a sphere. You see that? And then you can kind of position them, and then if you want cylinders to kind of go between them, basically once we've made one atom and one bond, after that it's about placing them, right? So then we could say add, and then we could go to mesh, and we could go to cylinder, and like that's obviously not a good shape for a bond, right? But if we just go to, and I'll help you all, you don't have to have this memorized, but if you look, if we go to, where is it? God, it's been a while. No, that is no. Yeah.
Oh,
Boy, this is a new version I just got, and I don't know where just the properties window is. Hello, properties window. Properties. Okay, well that doesn't matter. We can go down to where is it? It would be geometry. Ah, there we are. .4. 0.2. Okay, so we could probably say like, okay, you might want to fight with it a bit, but like, is that roughly what we want a bond to look like? Probably, right?
Yeah.
And then it's just a matter of you go like, okay, well now I've got an atom and a bond, right? And you can do the same thing here, where you could say like, if you want it, you could say, well, if you want, say say you've got oxygen, you want it to be slightly bigger, and just set the scale on all of them to 1.2, right? And then maybe to help you keep track of them, you can go to the materials website. Where's materials? God, I'm out of.
So if you have to make another one, just make it linear and then put 2.
Yeah, and so then we can just start copying and say, okay, give me another one of these, and just grab them, and then you can be like, okay, rotate them, right, like that.
Is there a way like you can kind of move the single ones, like when it's like when you print it, it's like kind of movable? Or
It
something?
probably will be a little bit flexible, yeah.
Okay.
But yeah, I wouldn't worry about it being. right? And so just like that. So I'll get you set up. I'll show you where to go, and we'll get files started for you. Yeah.
So my project is on two sides. Can I work on it on my phone?
I mean, you have the questions, and then it's like, you'll have obviously the name of the frog, and if you have a separate rulebook, like, you'll put like, I would say like page number or something. He's in the background.
6 anyways.
Speaking.
For the bacteria, like for photos and all that, we just have to make
Can
sure.
somebody get the lights for me, please, by the way?
We just have to make sure they're licensable, right? We
That's
don't
right.
have to, like,
Yeah.
source them.
No. I do want you to source them. I want you to source everything.
Okay.
Yeah, I want to know where you got everything.
My one looks like a mess, but this is like I'm gonna fix up the paint.
Hello?
But I only have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I have 10 questions, everything.
That makes perfect sense.
But does that look good? I'll just like
That's great.
and make it.
By the way, you saw those real big paper and hardboard over there if you want to use it. Take a look. You're welcome to it. You don't need to.
I
Like
might
behind
look at it.
Saphira.
I might look at it while we're leaving.
Sure.
The only problem is I walk 30 minutes, so
Well, you can look around here if you want, but yeah.
I probably want to bring it home so I can eat.
Right.
And so that, like, I just have everything down here. All the little messed up things I